Good afternoon, everyone, and welcome to this week's Ecology Live. I'm Pete Manning. I'm the chair of the BES's Events Committee, and also coming to you live today from the Senckenberg Biodiversity and Climate Change Research Centre in Frankfurt, Germany. Before we start, I'd just like to thank our sponsors, NHBS. If you don't know NHBS, then they provide books and equipment to ecologists and wildlife professionals. And as part of this sponsorship deal, they have an offer for free shipment to and free delivery for European customers until the end of August. And there'll be more details about that at the end of the talk. So also before we start, I just want to say a little bit about some of the activities that the BES does. As I'm sure many of you are aware, the BES is involved in all kinds of events and meetings. And one of those is the Quantitative Ecology Virtual Conference. This is going to be an online conference held in July with 4 days of talks and discussions and socials. And you can register for that, but the deadline for this is on Monday. So if you want to go and visit this meeting, attend this meeting, then you need to register before Monday's deadline. And you can do that at www.britishecological.society.org events. So now I'm going to introduce this week's speaker, which is Franciska de Vries from the University of Amsterdam. Before I hand over to her, I just want to say a little bit about how the talk works. We're looking at a 25-minute talk in which we're going to record it and post the video up onto YouTube if you want to watch it later. If you want to ask questions in the talk, then there's a little box down at the bottom in which you can ask a question. We'll select a couple of those questions, or maybe some more if we have time towards the end, and ask those. And then it'd be possible to address some of those questions on YouTube comments as well that didn't get asked in the actual session today. So with that, I think we'll hand over to Franciska, who's going to talk to us about Machiavellian microbes, which is a very intriguing title, and I'm really looking forward to seeing the talk. So over to you, Franciska. Thank you.
Great, thanks, Pete. I'm extremely pleased to be giving one of these talks in this very impressive seminar series of the BES. So I'm going to talk about soil microbes and why it's important to understand their response to climate change. Well, climate change is increasing droughts, and I guess you all remember the extremely dry year of 2018. When actually large parts of Europe and also of the world were experiencing an extremely dry summer. And these are just some British newspapers actually, but here you can see an image of my street in Manchester in the northwest of England. And I can tell you, this is not what Manchester normally looks like. So it was extremely dry, and it's very obvious what the consequences are above ground of these droughts. But we have this vast, incredible diversity of organisms living below ground, and we can't see them, and we also can't see how they respond to drought. But they perform very important functions for ecosystems. They decompose organic matter and they liberate nutrients for plant growth, and they're also all connected, so they work together to perform these processes. And so let's just actually have a look where these organisms live and how drought might affect these organisms. Because when we have a look below ground, we can actually see that these organisms, they're called soil organisms, but actually they live in the water because they live in the water films that are formed around soil particles, around mineral particles and organic matter. So nematodes and protozoa and amoebae, and bacteria, they are all actually living in this water film. So when the soil dries out, you can imagine that they don't like it, and they either become dormant or they actually die. So a lot of these organisms are aquatic, so they are really quite vulnerable to these droughts, but some organisms can cope better than others in the soil. And so what we actually know from a lot of previous work is that it's the sort of the more slow growing organisms, and especially fungal hyphae, that are actually quite well to cope with drought. So what we know is that these fungal hyphae, because of their thick cell walls, and because of their slower growth and their ability to access spatially separated water and carbon, for example, that they can actually cope quite well with this drought. And also that gram-positive bacteria are better able to cope with drought than gram-negative bacteria, for example, because of their thicker cell walls. What we also know from previous work is that bacteria and the organisms that feed on bacteria, because of their fast growth strategies, their opportunistic growth strategies, they can actually recover quite quickly after drought. So we know quite a lot about how these soil organisms respond to drought, but this is all from sort of independent studies focusing, for example, on a particular system or in a pot experiment, for example. So what we really wanted to know in this project, that I'm first going to show you some really hot off the press results from, is whether we can actually generalize the response of soil microbial communities to extreme events that are predicted to increase with climate change. So this is a very big project that was led by my postdoc Oceaan, who you can see here on the photo. And a lot of people are involved in this project: Chris Knight, Carolyn Weiser, Rob Griffiths, and Holly Langridge. And what we did is we went and collaborated with many people across Europe, and we went to 10 different countries, and we sampled the soils all across Europe. Actually, I say we went, but it was Oceaan who went to all these amazing places, and she took soil samples from 10 different countries. And actually, when I was writing this grant proposal, I was dreaming of all the amazing sites I would see. And in the end, I had a baby, and that baby would not drink from a bottle, so I wasn't actually able to do any of the field work. So it was Oceaan who did all this. So she went to all these different countries and she collected soil samples. And she did this during the growing season of 2018. And we also really attempted to do this sort of phenologically, so that in each site we were collecting soil samples at sort of the peak of the growing season. Obviously, this was logistically quite a difficult effort. So there were some hiccups, but all these samples were collected, and then these were transported to Manchester, and there we did a controlled experiment where we subjected all these different soils from 10 different countries and 30 different sites in total. We subjected them after a week of acclimatization in controlled growth chambers to a disturbance. And so we had a control treatment, and we had 4 different types of disturbance. We had freezing, heat wave, drought, and flooding. And so these treatments were imposed for 2 weeks, and then we had a recovery period of 1 month. And so of these soils, we took samples at the end of the disturbance, 1 day after ending the disturbance, 1 week, and 1 month. And by doing so, we could really sort of see the dynamics of the microbial communities, but also of soil properties and soil functions. But as I said, here I'm going to focus on how the microbial communities responded. And really, Oceaan has been working very hard to show you the first sort of snapshot of these results, because we only really got the data a couple of weeks ago. So this is how the community structure for bacteria, amplicon sequencing of the 16S gene, and fungi on the right side, how these communities were affected by the different countries. And you can see very clearly here that especially bacterial communities clustered together very strongly for the different countries, and also for fungi, we can see the same pattern. So this is very obvious. Okay, and then I'm going to show you this, the same graph, but then the treatment effect. So here you can see the different colors for the different treatments, again for the bacterial communities, for the fungal communities, and that's really a lot less obvious, right? So it's very clear that the differences in these communities that are caused by their different locations that they were taken from, their different climates, their different soil types, are far more important than this disturbance. However, the disturbance does cause a significant shift in community composition for both bacteria and fungi. And this is a little hard to see on this figure, although you can see in particular the pink dots separating out which is the heat wave, but I'll show you this in a different way in the next slide. So this is looking at the Simpson's index, again for bacteria on the left side and fungi on the right side. And here I'm showing you how this diversity responds to these different treatments. So this is actually the modeled results from mixed effects models. And so for bacteria on the left, for both fungi and bacteria, the control treatment is the red line. And then during recovery, after ending these disturbances, you can see what happened for the different treatments. So the control is the red line for both. And what you can really see here is, in particular for bacteria, is that this heat wave has quite a large initial effect, but also it just goes worse. It turns out worse and worse. So this is not, here the bacterial communities, the bacterial richness, not recovering at all. For fungi, we see the same actually for the freezing treatment, has a very big effect on fungal richness. But in contrast, in both bacteria and fungi, you can see that the drought, which is that sort of yellow greenish line, does really not influence the richness that much. Now, when we look at this in a different way, so what we did was we did an indicator species analysis for both bacterial and fungal communities. And here I'm showing you how many indicator species we found for the different treatments. So indicator species are species that respond very strongly to a different treatment, so that are very strongly associated with the change in a community of a specific treatment. And so again, what you can see here in particular, what I want to highlight is that actually flooding and heat wave, there are a lot of species associated with that change in bacterial community with those treatments. And for fungi, again, actually most species associated with heating. So there's a lot of species that increase with this heating treatment. However, what we also found, looking at these species that really change a lot, is that actually all the species associated with the treatments except heat wave, there were a lot of indicator species for those. So essentially this means that those species associated with all treatments except the heat wave treatment, that these species decreased in the heat wave treatment. So really, for both bacterial and fungal community, it was the heat wave that had the strongest effect. But obviously, this is just one way of looking at these communities, and we really have a lot more work to do in analyzing these communities, and in particular, to also link them to changes in ecosystem functioning. But right now, it's already very clear that it was actually heat wave that has a very strong effect here on the soil bacterial and fungal communities. And this is sort of in contrast what we expected, because we expected drought to be the most severe disturbance because these are mostly aquatic organisms. However, it's also very important to keep in mind that these samples were taken in that extremely dry year of 2018. And so that actually maybe these communities were already adapted to drought. So that's responses of microbial communities and whether we can actually generalize them. But in this part, I very sort of strategically did not mention that root that's here in the soil. And most soils have plants growing in them. So in this first part, I've been ignoring the plants, but roots and microbes really interact in a variety of ways, which can all be affected by drought. Because roots, well, they take up water, but also roots and microbes both compete for nutrients. Roots can collaborate with mycorrhizal fungi. Roots pump carbon into the soil, and they communicate with microbes through their exudates. Microbes can collaborate with plant roots, but they can also attack plant roots as pathogens, and microbes mineralize nutrients for plant uptake. So all these interactions, they might be affected by drought. And so we have to actually consider these if we want to realistically understand how microbial communities respond to drought. So the second part of my talk really focuses on how plants can modify how microbial communities respond to drought and what the consequences are of those changes, particularly for plant growth and plant community composition. So here I'm going to present you a very big mesocosm experiment, as you can see here, where we had big pots and we covered these pots with roofs. We simulated an extreme drought. And then, as in the first experiment, we looked at how the microbial communities and the plant communities were affected and how they recovered after drought. So we used plant communities that represented a gradient of dominance of 4 different species. So all communities included these species, included all 4 species, but in varying abundances. So we had a fast-growing grass in there, a slow-growing grass, Anthoxanthum odoratum. We had 2 forbs, a fast-growing forb, Rumex acetosa, and a slow-growing forb, Leontodon hispidus. And so these plants also differed very strongly in their root systems, as you can see here. So we subjected those plant communities to drought, and then we looked below ground how the fungal and the bacterial communities responded. So here you can see again the richness and the evenness for bacterial and fungal communities, bacterial at the top and fungal at the bottom. And what you can here clearly see that over time, so the gray bars here represent the drought, that actually the fungal communities responded quite strongly and positively to the drought, but recovered very quickly. Whereas what you can see here is that these bacterial communities, they did not really recover. And this is really quite in contrast to what we were expecting, because we always thought bacterial communities recover quickly. Then what I also did is I looked at the co-occurrence networks in these communities. And here I'm showing you only the co-occurrence networks for the late recovery sampling, which was 2 months after this drought ended. What's very clear here is that these fungal networks, they were really not very much affected by this drought. They also look very different from the bacterial networks. The bacterial networks are much more connected. But in particular, what you can see here is that 2 months after that drought had ended, this bacterial network was really strongly connected, which might mean that these bacterial taxa, these species, are strongly co-oscillating. They're responding in the same way, which means that they might not be very stable, that the drought is really affecting how they're responding together. So, but I've ignored the above ground so far. So what we also saw at this late recovery sampling, so 2 months after the drought, is that it was this fast-growing grass, Dactylus glomerata, that was really increasing in abundance in response to drought. And you can see that here in this PCA, that's the red dots that were associated with an increase in Dactylus glomerata. And that actually, in this SEM here, you can see that it was the increase in this fast-growing grass that actually, through increasing plant growth, it actually depleted soil moisture, even still 2 months after this drought was ended, because it was evapotranspirating and growing a lot. And that then still affected both fungal and bacterial community composition, but bacterial community composition much stronger than fungal community composition. And also it directly affected bacterial community composition. So not only through its effect on soil moisture, its continuing effect on soil moisture, but it also directly continued to affect bacterial community composition and actually preventing that from going back to where it was. So we also found in a follow-up experiment that these changes in plant communities and microbial communities affected the future performance of the plant species in this experiment. So what we did is we used the soil from plant communities that were dominated by this fast-growing grass, Dactylus glomerata, from both drought and control soils, and the soil from plant communities dominated by Leontodon. And then we had both species growing in their own soil, so in the soil that was conditioned by this same plant species, but then we also grew it in the soil that was conditioned by the other plant species. And so by comparing how it grows in its own soil to how it grows in the foreign soil, we can see whether it positively or negatively affects its own growth, and whether that was affected by drought. So what we found here, you can see whether this feedback on itself was positive or negative. So when these bars are above the 0 line, the plant positively affected its own growth. When it was below, it negatively affected its own growth. So what you can particularly see here was that this fast, slow-growing forb, the ontodon, positively affected its own growth under control conditions, but in drought soil, it actually negatively affected its own growth, which means that its own abundance was going down in this future experiment. Now, we also looked at how they were growing in competition when they were growing together, and how they responded to being grown in their own soil or in the foreign soil. And again, we found actually that Dactylus, it seemed, was affecting its own performance more positively in its own soil. So these changes in these soil microbial communities, they were actually affecting future plant growth, and this direction changed when there had been a drought in these soils. And this might mean that actually some species might positively and some species might negatively affect our own future performance in response to drought. Now, the next question was whether we can understand what the mechanisms are through which these plants affect the microbial communities. And so this is really the last part of my talk, because what we wanted to know was whether there is a role for root exudates in driving the response of these soil microbial communities to drought. So we looked at 2 species here, Holcus lanatus, which is again a fast-growing grass, and the same forb that was in this pot experiment, Rumex acetosa, with very different root systems. So again, we imposed a drought on these 2 species, and then we had an end of drought sampling, but also a sampling after the 2-week recovery. And here I'm going to focus on this sampling after these plants had recovered for 2 weeks. So what we did was we harvested these plants that had been recovering from drought, and we compared them to plants that hadn't had a drought. So for both species, control and drought, we sampled the soil and we collected the root exudates. And if you really want to know how we did that, then I'd be happy to also answer some questions in the future. But we harvested the soils and the exudates. And then what we did is we reciprocally added all these exudates from these individual plants, whether they had had a drought or not, to all these soils that had been conditioned by these individual plants and had experienced a drought or not. And so this was done in microplates, micro-rest plates, as you can see here. And this was a lot of pipetting. This was done by my postdoc, Angela Straathoff. This was around 6,000 individual wells of pipetting, I think. So, but by doing this, we could really disentangle the importance of the history of the soil versus the history of the exudate in driving microbial respiration. So this is what we found for the soil. So here you can see the soil history. So whether the soil had a Holcus plant or a Rumex plant growing in it, and whether the soil had experienced a control or a drought. And then this shows the respiration coming from that soil. And these are actually the individual soils in pairs for block, pairs of control and drought soils. So yeah, I mean, I guess you're just wondering what's going on here. Well, nothing. So the soil, whether that soil had a Holcus or a Rumex growing in it, or whether that soil had had a drought, did absolutely not matter for the respiration coming from that soil. Now we're going to have a look at the root exudates that were added to the soil. Oh yeah, and I forgot to say, actually, these soils were changed in response to drought because the microbial communities have changed quite strongly, but this did not affect the respiration in that soil. So now we have a look at the role of the root exudates. So this is exudates that were coming from Holcus plants and exudates that we collected from Rumex plants. And here you can very, very clearly see that even the individual exudates collected from individual plants mattered for the respiration coming from that soil. And you can also see that both for exudates collected from Holcus and exudates collected from Rumex, that the droughted exudates, so the exudates collected from plants that had experienced a drought, triggered far more respiration than the exudates collected from plants that hadn't experienced a drought. And this is 2 weeks after recovery. And so these plants still clearly have some sort of physiological adaptation to this drought that they'd experienced. So they exude exudates that trigger the microbial community to be more active. And so what we hypothesize is that this might actually be to stimulate the microbial community to increase nutrient mineralization and maybe facilitate quick regrowth after the drought. So this might actually be a mechanism through which plants promote their own abundance after a drought, and might actually explain why we saw a higher abundance of, for example, that fast-growing grass in that previous experiment, and that species actually feeds back positively to its own performance after a drought has ended. So just to summarize part 2, root-microbial interactions are really important to consider if you want to predict ecosystem responses to drought, because they might actually underlie the things that you see above ground. So they might actually, certain plants might use their microbes to promote their own abundance after a drought has ended. And this is also something that we propose we might be able to use, for example, in agricultural systems. And so this is here we hypothesize in this figure from a recent paper that was published in Science this year, is that actually during a drought, there are interactions between plants and microbes that might help the plant cope with that drought. But when you have a very strong or long-term drought, actually everything stops and the plant and part of the plants might die and microbes might die. But then when the drought is ended, that plants might employ specific mechanisms, for example, through altered root exudation, where they actually use those microbes to recover from the drought and potentially promote their own future abundance after a drought has ended. So that's really what I wanted to tell you today. But obviously a talk is never complete without acknowledging really everyone who's worked on these data. And so that's really a lot of people, also a lot of collaborators across Europe. And this work was funded by NERC and the BBSSE, and also the European project EcoFinders.
Thank you, Franciska. So thanks a lot for that. We're just going to ask a couple of questions now. I think we've got time for that. I'm going to choose some which were asked by several people, methodological questions. One of these was for the heat wave. A lot of people were interested in how hot it actually was.
Yeah, it was either 30 or 35 degrees. But it's also very important to say that actually the soil moisture was obviously kept constant at the same level as the control. So quite often the effects of a heat wave also causes drought, obviously. But here these disturbances were really sort of independent of each other.
Thanks. Yeah, and the other question, a methodological one, a lot of people were asking was, how do you collect these root exudates? And what's the method for that?
Well, we're hoping to submit a methods paper fairly soon. But what we've done, I think, actually in past years, people were inclined to collect them in sterile systems. But we would argue, and I think people are also increasingly recognizing that this is obviously not a realistic representation of the field, because in the soil you have roots growing in the soil, and the root structure is very much influenced by the soil, but also there's interactions with the microbes, and these all affect root exudation. So what we do is we grow the plant in soil, and then we wash the plant, but we let the roots repair themselves before we collect the root exudate, as to avoid any sort of root damage. So you're essentially collecting cell contents if you collect the root exudates immediately after washing. So we let the plant recover, and then we collect the root exudates. And what I found quite striking was that we saw that signal of that drought very strongly still 2 weeks after that drought has ended. So.
Okay, thanks. Yeah, and I think we'll just finish with one question, which is a bit more broad. This comes from Krishna Girish, and they were asking, are these microbes that you see in the soil, if they're aquatic organisms, are they the same as what you see in an aquatic environment? And if they're not, then are they different? And what is it about them that's different? Do they prefer the higher resource availability in the soil? Or can they not compete in aquatic environments?
Yeah, it's really, really interesting, actually. Some are the same, yeah, but mostly they're different because of the very different sort of more heterogeneous structure of soil and more nutrient and carbon rich. Yeah.
Mm-hmm. Thanks. Yeah, and then just one final question. If this, let me just find it amongst all the many that have come in. We will put these up on YouTube so you can see them. This one was from Isabel Alonso, and they were asking if there's anything a land manager can do to actually influence these mechanisms that you've been describing.
You mean in a, yeah, well, so what we're proposing on that sort of final slide is that actually we might be able to use these interactions in future agricultural systems. For example, if we are able to pinpoint the genes that control root exudation, for example, or pinpoint the genes that influence roots, like root growth, how it's sort of spatially distributed over the soil. And I think a lot of work is being done on that at the moment, actually. But we might also be able to, for example, add specific microbes to the soil, or prebiotics or probiotics. But I mean, I acknowledge that this is actually still all quite far off, actually, because what we're doing is really the fundamental research in these interactions.
Absolutely. Okay, so with that, I think we have to finish for today, but thanks again for this really great talk. I really enjoyed it. I hope you all did too. Thanks for the many hundreds of people that joined online. Before we finish, I just wanted to say a few words about next week's talk. And that is by Fidisoa Razam Bain Arivo, and he's going to be talking about shared bugs, microbial sharing network among the carnivores of Madagascar. So I hope you're all looking forward to that. I know I am, and I'll hopefully see you all next week. Just a final few words about the talk. This week's talk will go up on YouTube, where you can see other Ecology Live talks and ask further questions, and we'll take the questions which we weren't able to address today and put them up on there. So with that, I just want to thank you. I'll leave you with the offer from NHBS, which is their free shipping on European orders until the end of August. I hope you take advantage of it, and I hope to see you all again next week. So thanks a lot from me, and see you all next week. Bye-bye.